Hey, welcome to summer internship in microbial DNA barcoding and barcode analysis. We in next 21 days, we are going to analyze various aspects of microbial DNA barcodes, or we are going to analyze various DNA fragments from microorganisms. So why this DNA sequences has become very important is in microbiology. Without DNA sequencing, you cannot declare a species identification. Okay, if at all you want to identify any microbial biological system to species level, you need to go for DNA sequencing, especially RPOB and 6' RNA are mandate for identification of a species. This is not something new, the barcoding in microbiology, because for last 45 years, we are dependent on DNA sequences for microbial species identification. Even Carl Woos classification of three kingdom concept, archaea, bacteria, and eukarya, is because of the DNA sequences. Okay, so what to expect in this internship? Before we discuss elaborately on the internship, I want to quickly introduce DNA Barcode Biotechnologies, which was founded in 2013, and they are a full-fledged DNA sequencing company, especially Sanger's DNA sequencing, and they are most affordable DNA sequence service provider in the country. I can vouch for that. I've been their customer for more than 15 years now, and they are not purely business-based company. I will also introduce the various initiatives they are doing. So this is a Megabase 1000 sequencer and PCR workstation. More than 27 pieces are there that can be controlled through a computer system. And this is the bread provider of the company, Mermaid 192X synthesizer. This is a primer synthesizer, PCR primers. In my knowledge, they are running for 10 years continuously. 24/7, no Sundays, Saturdays, they will be keep on running. They are the bread provider, like I say. They are providing primers for 18 rupees per nucleotide base. To synthesize one nucleotide base, they are charging 18 rupees. I think it's considerable rate. And here is a ABI 3700 model, Sanger sequencer. They are also running parallelly. Along with this, they also have Illumina HiSec, MiSec platforms. All right. So the internship, this is all about various objectives like understanding the concepts and importance of DNA barcoding, learning about the key molecular and taxonomic markers, 6' RNA, RPOB. In case of fungi, it is ITS. We will learn in detail about it, and we will gain hands-on experience in sequence analysis. This is something very important, and it's a prime objective of the internship to provide a hands-on experience, so that tomorrow when you sequence your own DNA, you should be in a position to interpret them, handle them, refine them, prepare them for analysis, submit in GenBank, get accession number. You should know what are the other, downstream processing after DNA sequencing. So develop skills to perform blast analysis and other DNA-related sequence analysis. And you should be independently able to perform microbial identification process. Thereby, you can run separate projects. Okay. So see why this microbial identification is very important is for any field, consider an industrial field, agricultural field, biofuel production, textiles, food processing. Identification of microorganism is something very basic. The research starts from there. Okay, it's not just an experiment or process. You can see it is a big market, which is worthy of around, it is projected to grow by nearly $5 billion in 2030. And if it is 8% growing, it will be around $5 billion by 2032. Okay, so it is an amazing field to pursue. If you want to, seek a career, I think DNA sequence analysis and sequencing process actually fetch you a lot of jobs. And this is a structure of 21-day program where we will be talking about fundamental analysis of DNA sequences in week 1. In week 2, we will be talking about little advanced analysis like phylogenetic analysis, nucleotide diversity determination. In week 3, I'm planning to do a mini projects, individual specific mini projects. So this internship will have lectures, demos, and hands-on videos given so that you can hear the videos through your mobile phones and practice them in the laptop. Okay, at the end of the training, everyone will be skilled in analyzing their own sequence data because we are providing two sequencing reaction for free. You can send us your sample, any sample, you will get your sequences. Using your sequences, you can do the analysis. So I urge you to send the samples within a week so that the company will deliver you the results on third day after receiving the sample. Okay, and meanwhile, these are the various online and offline tools you will be learning in this internship. So today's the day one's agenda is just to give an what is barcoding and why it is important, and what are those barcode genes, and understanding the workflow of what is involved in barcoding. Then setting up the practical sessions in forthcoming days. So what is barcoding? Like you have a universal product code, UPC codes in the product that describe the entire history of the product. Every organism have a DNA sequence which can be read and identify the species. In other words, barcode genes are species specific. The similarities and variation between the DNA sequence is very less among the species, meaning every species have the unique barcodes. Okay, so once we just sequence them and put it in the database, even unknown samples now can be sequenced for that particular region, compare it with the database, and you can identify the species. You may not require the experts in that case of identification, but experts are required for synthesis of the database. I will explain you what is that. He is an expert and visionary who came up with this idea called DNA barcoding. He is an entomologist. He started with butterflies, and his idea caught up like a forest fire. Even in India, we have a Zoological Survey of India and Botanical Survey of India taking lead in DNA barcoding works. And we have various other organizations actually contributing to the DNA barcodes. Not only geographically, even phylum-wise. We have fish barcoding, spider barcode, Lepidoptera barcodes, even plant barcoding groups. Every phyla has their own special groups working on them. All started in 2003 when this paper came out, biological identification through DNA barcodes. From that, they realized they initially proposed cytochrome C oxidase subunit 1 for most of the phyla, but they realized that it may not work in plants. So for plants, they proposed RBCL and MATK. Again, realization came during February 2012 that it will not work in the fungi. So ITS region has been proposed as a barcode for fungi. And here is one of my paper in next year after this proposal, 2013. I tested this ITS region in other marine fungal that has been collected along the coastal regions. So what are these primary barcodes? For fungal identification, it is an ITS region. You see, for bacteria and archaea prokaryotes, we will say 6' rRNA gene. For fungi, we will not say gene, we will say region. Why? Because this internal transcribed spacer 1 is actually divided, separated from spacer 2 by 5.8 S gene, which the whole region is flanked by small subunit RNA, 18S rRNA and 28S rRNA. So this entire region segments are called ITS region. And ITS gene is the, ITS region is the barcode for marine fungi. When I say fungi, it's a common word. You should know we also have molds and yeasts, the different forms of fungal growth. Okay. See, when a particular gene has so many primers developed, you should know the importance of this gene in the work, right? You see, here we have shown a primer map. Wherever you see a bold, this means that these regions are explored for metabarcoding. Primers are developed from these regions for metabarcoding. You should know even in the ITS we have a variable region and conserved region. Usually from the conserved region, the primers are developed. So the bolds are actually from metabarcoding. Wherever you see underlined primer region, these are actually specific fungi. If you want to determine Aspergillus niger alone, they have designed a primer from this region. So underlined are primer regions of specific fungi. And all the red color are actually the primer regions for global microbiome project. And all the purple color are actually a primer region for glomeromycota. And all the blue color you are seeing in the primer map is actually for Earth microbiome project. You should know when you have these many primers. You also have a universal primer that will work for all fungi. Okay, these many primers developed. You should know the importance of this region, ITS region. Why it is so important? Just take a look of these two mushrooms in the left-hand side. One is actually Marismius imitarius, and other is Marismius bambusiniformis. You should see in ordinary people's eye, it looks exactly similar. Right? See this structure and see this structure, exactly similar. But when you sequence them for ITS region, they have done it in triplicates, three mushrooms. You see in the phylogram where they are placed, they are two different species for sure. Okay. So this is why we need DNA barcoding. For a conventional taxonomist mycologist, this will be very much challenging. Not only this, you see here, the spore morphology, ascocarp morphology plays a vital role in identification. These are different, looks like different types of ascocarps, actually are sampled from the same strain. Okay. When a single strain of mushroom have a different ascocarp morphology, imagine the difficulty a conventional mycologist will face. But see, when we sequence, when this author sequence various forms of ascocarp for ITS gene and produce this phylogram, all are cladding in same branch, meaning they all belong to same species. Okay. So similarly here in another example, the same results. Okay. So this is ITS region, and for prokaryotes, we have a 6S rRNA. Does that mean it is a perfect barcode? I would argue because there is something called RPOB, RNA polymerase beta subunit gene, which is actually highly conserved, present in all bacteria. They are housekeeping genes, okay, and they are around 3,500 500 base pair in length, and they are much more conserved for bacteria. See, for any gene to act as a barcode, they should have a relatively lower intra-species variation when compared to interspecies variation. What is this intra-species variation within the species of same genera or the strains, various strains among same species? They are just intra-species, and various species in a same genera in one single genera is an interspecies. So this interspecies variation should be higher than this intraspecies variation. First of all, what is this variation? When you say, when you ask for variations, when you compare a two DNA fragment, say for example 100 base pair in length, 100 base pair nucleotide in length, when you compare these two DNA sequences, if the DNA sequences differ in two nucleotides, then I would say it has 2% variation. So whenever we say percent, we are calculating it for 100. So when you take 100 nucleotide as a parameter, we can easily calculate the variation. So here, nucleotide diversity is given, and the length of the gene. 6S rRNA is usually 1,500 base pair. This is 3,500 base pair. See the nucleotide diversity. In our internship, we will also calculate how to, we also say how to calculate the nucleotide diversity. You see the variations. So this is beautiful, right? So that's why RPOB is considered as a much more better gene to resolve even species level or zero or level diversity, but still 6S rRNA is used because simply because of convenience. Okay, we have flooded the database with 6S rRNA for last 20 decades. I'm sorry, 2 decades, 20 years. But now, they are for every species. If you want to declare it as a new species or a species, if you want to identify a species, you are sequencing 6S rRNA. We have millions of copies in the GenBank and other international nucleotide databases. If you want to use RPOB now, you have to spend more to generate such a data, which is practically impossible. That's why still we are sticking on with RPOB, because for barcoding to work, you need to have a reference sequence in the database. If you don't have a reference sequence, that's the limitation of barcoding. Okay. And during this lecture, I will also tell you how this barcode biotechnology is contributing to enrich the local library with various species. So regarding the microbial barcoding workflow, if since you guys are sending the samples, 2 samples to the company, this is how we are going to process a sample. Once we receive the sample, we hardly take 10 milligram or less of your DNA of your bacteria. In a loop, we have something called colony PCR, where we will heat the double distilled water at 80 degrees Celsius. We will pick a small portion of bacteria, dissolved in the double distilled water, maybe 0.1 ml will do, 100 microliters will do. Then again, we will heat it to 80 degree for 10 minutes. Between every 2 minutes, you can vortex the sample, and then make sure the DNA is released. And we also making sure the endonucleus of bacteria did not degrade the DNA. Okay, so that's why the 80-degree treatment. From there, when master mix everything is ready for the PCR, we will just pipette out 1 microliter or 2 microliter from the slurry, directly put it in the PCR master mix, and we'll go for PCR. Okay, we will be using a universal primers of 6S and ITS for bacteria and fungi respectively. So once the PCR is done, we'll check it in agarose gel. When we will use 10 microliters for 25 microliters PCR. We use 10 microliters to check it in the gel. When we see a sharp band, we will split the sample into two. We will go for single standard amplification again. Then for every sample, two reactions, forward and reverse, will be amplified and it will be fed into the ABI model 3700 XL model, and the sequences are produced in form of chromatogram. Our internship starts here. From chromatogram, what are the analyses we can do? Okay. So this is how the chromatogram typically looks. This is an ABI 3500 XL model. So they are actually attached to the computer, and where the chromatogram files are stored there. We will have a session on how to refine the chromatogram, extract sequences, how to name and classify the peaks in the chromatogram. Okay. Then after taking the sequences, we will see how to calculate the nucleotide genetic distance and how to prepare a species identification report, including merging the forward and reverse reactions, the single strand PCR we have did during the after gel checking the PCR replicon. How to merge them, how to form a single sequence, how to subject that to blast analysis so that we will know the species, how to interpret the blast results. After that, how to multiple sequence align them, our sequence along with the reference sequences we collected from the blast, how to draw the phylogram. See, phylogram are graphical representation of similarities and variation in the DNA sequence. What is that? See, sequences sharing very less variability, or they are more similar, highly similar sequences are branched in the same clade in the phylogram. So see the phylogram from left to right. Okay. Initially, there are two branches. From this branch, we will call them as a node. Okay. From this node, we have two more branches. From this node, two more branches. This leuconostoc lactis and leuconostoc palmae actually are very genetically closer. That's why they are placed in the same branch. When compared to leuconostoc mesenteroids, they are placed away from this. Okay, so those who are very similar, have high similarity, are placed in the same branch. Those who are very dissimilar, has more variability, are placed away from each other. This is a very basic way to interpret the phylogram. Okay. Not only that, they are, I told you, they are the they are the actually graphical representation of similarities and variation. How similar, how variable we can calculate using genetic distance data. Actually, Mega calculate the multiple sequence alignment file to genetic distance data, and based on this data, these phylograms are plotted. Okay, we will also learn how to prepare this data using molecular evolutionary phylogenetic genetic analysis software. Okay, so this is how you will be receiving your sequencing report. You should know how to prepare the report, how to read the report. We will also see how to submit the sequences to GenBank and get the accession numbers. Most importantly, we will calculate barcoding gap. What is this barcoding gap? See, for bacteria, the barcoding gap is 3%. What it means is, when I do the blast analysis, if the bacteria have less than 3% variation or more than 97% similarity, they can be considered as same species. Okay, when you do the blast analysis, if the E. coli shares 97% or 98% similarity with an E. coli, the sequence shares 98% with E. coli, it is E. coli. If it shares 96% with E. coli, it is Escherichia, not coli. Okay, it may be Escherichia faecalis or some other species, because the species cutoff is 3%. If anything you want to declare as the same species, they should be either equal to 97 or more than 97. Okay, so the genus cutoff is 5%, that is 95 or more than 95% similarity. Family cutoff is 93. Order, class, we have various levels of cutoff. If you do not have a levels of cutoff, better you propose it for your species, because that will be considered as new data which can be advocated when you communicate your manuscript. Okay, so what are the expectations from us to, again, towards you? Please use mobile phone or any other device. You should have two devices for this bioinformatics internship. Please hear the videos through one device, mobile phone or anything using AirPod, and simultaneously do it with the other device. That's how you can learn. Actively engage during the discussion part and complete daily assignments. Every day, we either have a live sessions or recorded sessions. Especially for hands-on training, I recommend recorded sessions, because everyone's rate of learning will be different. Internet speed, the environment which you will be using to understand these videos, everything is highly variable. So if you have a recorded session, you can pause it anytime you want. You can play it at your convenience. which is very much effective in terms of learning new concepts. Okay, so I advocate for pre-recorded sessions, and everyday assignment will be given, and your insights and challenges can be shared via the daily reflection form, which will be shared in the WhatsApp group. You can record your feedback every day so that we will get back to you whenever we could. Okay, and from tomorrow onwards, we will start with assignments and see these are the various services provided by Barcode Biotechnology. You even can send a mixed culture sample if you have a poor microbiological infrastructure where you cannot or you lack skills, you cannot spend more time on refining pure culture. You can simply indicate which colony you want to purify to us, send the plate. We will do the purification, sequencing, identify the species, prepare the glycerol stock. We will give it back to you as a pure culture. Okay, apart from identification. And we are also doing archival identification. In this internship, you have two sequences for free. Either you can select two different species and get it sequenced, or one species, two different genes. For example, if you are working in a bacteria or archaea, you can point to us to sequence both 6ness and RPUB, which is very good. Even for fungi, we can target 18ness and ITS. This is a service they are offering. For mushrooms, 18ness and ITS can be targeted. This is multi-locus gene sequencing. Okay, we can send the sample. You see, the worth of I know people who are doing sequencing for a long time know the per sequence cost is around 4,500 in the market. Okay, so we are giving two sequences for free. Now you understand the rate of why the internship have a fee. Actually, there is no fee for learning new concept. It's all for the sequencing. Okay. So Forge identification also we do. You can simply send us the Forge blocks. We can excise the buttons and we can go for identification. Insect barcoding, you can simply send a animal tissue sample in 70% ethanol. We will barcode cytochrome C oxidase, one gene for identification of insects, animals, plants. For plants, we have RBCL. For animals, fishes, insects, copepods, lizards, squids, any animal sample you can send. Even community sample. You can send a group of animals. We can extract the entire DNA of the community and pinpointedly sequence or target few species and sequence them. It's very much possible. Even in forensics these days, you know, when you are going for forest survey of animals, even fecal samples you can send us. We are actually isolating the DNA and we can tell you the host DNA, what is the species, because the epithelial cells are also shared along with the fecals. And also we can decode the dietary composition. If you tell them it's a carnivore or herbivore, we can use a specific plant or animal primers to decode what kind of diet they have consumed. This is very much possible for birds, and many species are actually threatened. You cannot sample them directly, but the fecal matter is very much easily, you guys can sample it. The dried fecal material, from that we can extract DNA and to, tell you what bird is this. We have ornithy of one and R1. It is very much specific to birds. For plants, we either target MATK or RBCL gene. You can send us a tissue sample or even the plant herbal powders. We can isolate DNA from them. And for even metabarcoding, you can simply filter the air using, trap them in the filter paper, decode the entire diversity from that. So Barcode Biotechnology, like I told, they are not just a business-based company. They run a initiative called, which I like it very much, called 1000 Barcode Initiative, I-1000. And this is all about barcoding 1,000 species that is yet to be barcoded, whose barcode gene sequence is not present in the database. Okay, so they are very much peculiar. I think they have already met the objective of 20%. They are trying to meet this objective through various other programs. Why this objective? In order to barcode to work better, you need better have every species sequence in the database so that even non-taxonomists can now identify the species. Why it is important? See here in this study that is published recently, 2 years back. The size of the bubble indicates the number of barcodes present. See the size in India, see in Canada. Ironically, you see countries with low biodiversity have more barcodes, and country with high biodiversities, you see the number of barcodes here. Okay, so that's why we need. I think it's because they are actually having a high-end technology, and countries with high biodiversity, we are just catching up. Okay, so you see, I just took 20 species. I googled it. I took the 20 species of which is endemic to India. and the species are listed in red color actually are not there in the database. If I just go to the NCBI and search for it, their sequence is not there. Meaning, if you can sequence them now, you can easily make one publication. Okay, so this is an example I will show you. Here is for endemic animals of India, and the species are listed in red color are not present in the database. When you sequence them, it is first time barcoded species. You can simply publish a paper like this. This is around 2 point something is my paper. I hardly spent 15 days sampling and sequencing and 15 days in writing. In next 15 days when I communicated, I got accepted. Okay. You can have this title something like first barcode subbathynomus or something like this. And similarly for this paper, one of my paper where we advocated various species boundaries. Okay, so we sampled around, we documented, we produced 171 barcodes for 54 fungal species, mold species. Okay, so I proposed a barcode gap of 15% for genera level, meaning for genus, it should be at least 80, 85 or more than that, similar to the database sequence. Then it belongs to same genera and we can think about species. Okay. For genus, family, order, class, phyla, I have done an analysis. How did I do? I produced these 171 barcodes and I mapped them with the closest relatives and prepared a pairwise distance data. Okay. Using Mega, we prepared something like this. Then we calculated it for a percentage and then we propose the barcode. Okay. You will be learning how to define a species here. And in this paper, I have produced around 157 marine yeast species from 1017 barcodes. Okay. And 13 species are actually barcoded for first time. It was not there in the database at all. So now publishing becomes very easy. Here is a proof that ITS is very much intra-specific. See, and I have used 5 different species of Cryptococcus. Still, ITS can clad the same species of Cryptococcus in same branch. Can you see here? That's what I have mapped it here. I have marked very clearly. This is another proof that ITS is working fine, both for molds and yeast. Okay. So there is a dedicated database called Barcode of Life database. If you want to see this yeast data set, you can visit this database. This is the project title, Marine Yeast of Indian Coast. You can search for this and you can see various barcodes here. Okay. In case of prokaryotes, that is bacteria and archaea, still this barcode gap is not defined. I just said that 3% can be used as a general cutoff, but for Salmonella enterica, they have their own cutoff. For pathogens, their cutoff is even narrower. They may have a 99% similarity with 1% variation, or even less than that. So for pathogens, it is very much important to have species level cutoff. We cannot have a general cutoff. This is where we can contribute something. If you are using your microbial sample or any samples, we can not only identify them, learn how to identify them, but also we will do a genetic distant analysis to propose a cutoff values for our species. That we will do in the third week of the mini project. So this is how we will calculate. We are learning actually slowly one by one. We will come to this point and we will see how to calculate barcode gap within different species in the same genera. So I just want to remind you, there are 7 different ways you can send the sample. You can even send any samples of animals, plants, plant leaves, environmental sample, mixed cultures. Even one microbial sample asks us to sequence 2 genes or send us 2 samples and get them identified. Okay. So there are various ways you can do it. Even I see many people, they may not have an access to the lab. I'm telling you a good idea. You can propose this to any other students who are actually working on microbial identification. You can tell them, I have 2 free sequencing. If you are willing to join me as a co-author in your paper, I can contribute 2 sequences to your project. We will publish the sequences. I also will contribute on sequence analysis. I can analyze and I will give you a blast result, phylogenetic tree, genetic distance analysis, all those data you can produce and contribute to their work so that you can be a co-author also. Okay, think about it. And that's it. Happy barcoding. Let's keep learning about the DNA sequence one at a time. We will make a progress. I hope everyone will be skilled, and we will see how to make a contribute a meaningful data at the end of third week. Thank you.